Great. So our second speaker today is Dr. Mike Guertin. Dr. Guertin conducted his PhD in the lab of John Liss at Cornell. From there, he moved to the National Cancer Institute in Maryland to conduct his postdoctoral work with Gordon Hager. Before in 2015, he began his own lab at the University of Virginia before moving to the University of Connecticut in 2021. Dr. Guertin has long demonstrated his commitment to diversity and inclusion, stemming from his time in the Air Force, helping colleagues access their educational benefits, as well as a passion for teaching. He recently became an instructor for the Cold Spring Harbor Gene Expression course. His research is characterized by rigorous investigation of transcription factors, especially nuclear receptors, transcription and stress responses, and chromatin structure and function. All this using molecular biology, genomics, and bioinformatics approaches to study these processes. I'll note too that his lab website has an array of computational analysis vignettes, helping make their bioinformatic tools accessible. So with that, I'll finish by I'm very excited to hear a seminar today on kinetic networks of differentiation cascades, identify key regulatory nodes and transcription factor functions. So please join me in giving a warm, albeit virtual, welcome to today's second speaker, Dr. Mike Guertin.
Thank you so much, Ben, and thank you to all the Fragile Nucleosome organizers. This is such a great series. Okay, so my group is at UConn right now, and I just want to make a plug for the Systems Biology Center that we're in. We are recruiting. We're recruiting hybrid labs that are hybrid computational and experimental, or just purely dry labs. We have a pretty decent gene regulatory network crew here, and it'd be great to build that. Okay, so I'll spare you the background about chromatin and transcription, and just briefly talk about two of my favorite Nobel Prizes, which I think of as transcription factor Nobel Prizes, and just highlight the fact that they're really question-driven science, and they're not overly hypothesis-driven. Of course, one can take any question and kind of articulate it into a hypothesis, but you can imagine when people in the 80s were mutating Drosophila, and the real question was, what genes are important for developmental patterning? And they had this beautiful, elegant screen. Classic genetics where you just perturb, observe, and map. The thing that's noteworthy for this talk is that over half of the genes that regulate patterning in Drosophila are transcription factors. So like things like antennapedia, where you would get the leg growing out where the antennae are supposed to be, or ultrabithorax, where you have an extra set of wings and extra thorax. More contemporary, but getting more and more dated as we age, is the work from Shinya Yamanaka, where the question was really like, at the time it was unknown, but can we even take differentiated cells and induce them to become pluripotent stem cells? And he had a really well-designed, elegant screen where it took 24 different genes that were overexpressed in embryonic stem cells versus differentiated cells, and was able to narrow that down to 4 transcription factors that can induce pluripotent stem cells, and we can obviously turn on different transcription regulatory programs to further differentiate these pluripotent stem cells. Okay, so what are the questions that are been driving our research as of late? One is which regulatory elements and transcription factors are important for differentiation cascades, or really any kind of regulatory cascade. And this isn't like the typical network that you would see on the cover of a high-profile journal 10 years ago, where you have all these different hairballs of nodes and edges, and each edge is some kind of ambiguous relationship between nodes. Here is a bipartite network, so it has 2 different flavors of nodes, where you have regulatory elements in the genome, you have genes in the genome, and if you have a directed edge from a regulatory element to a gene, that's an inferred cis-regulatory event. So saying that binding of factors at this regulatory element is, if you can see my cursor, binding factors at this regulatory element is regulating this gene. This gene, in fact, encodes a transcription factor. It binds to a bunch of different places in the genome. Some of them are terminal leaf nodes, so there's no evidence that they're regulating anything downstream. In some cases, binding correlates with the change in transcription of local genes. And this transcriptional cascade can propagate through nodes that are transcription factors that are binding and turning on things. So can we identify the key factors and regulatory elements in a way that's as unbiased as possible? Another question that's been driving my research program for many years are what are the key steps in transcription that sequence-specific transcription factors regulate? As you all know, cofactors or general transcription factors tend to specialize in any one of these steps in transcription, but none of them have inherent sequence specificity. So they are recruited by sequence-specific transcription factors, kind of highlighted here, usually ignored on these transcription cycle illustrations. But transcription factors themselves tend to specialize because they have higher affinities for some cofactors versus others, and they compete with other factors for certain cofactors that maybe are involved in remodeling or pause release. And I think we need to start thinking about, many of us have transcription factors not as activators as or repressors, but what mechanistically do they do? Like, do they recruit nucleosome remodeling complexes, and maybe those would be considered pioneer factors, or what people have been calling pioneer factors, or do they tend to catalyze or recruit factors that catalyze these other steps? And all this work is performed by Arun Dutta, shown here in this dated photo of my lab. And he'll be defending his work next month. Okay, so the model that we chose to study this is a well characterized model of adipogenesis in mouse 3T3L1 cells. In short, you can take these cells, you add a glucocorticoid, a CAMP agonist, and insulin, and then 6 days later, these fibroblasts will become adipocytes. And a lot of stuff happens in 6 days, and we wanted to track that stuff as unbiased as we could. So here's the experimental design. We have your pre-adipocyte fibroblast, and over 7 time points over the first 4 hours of adding this cocktail of drugs, we did ATAC-seq and PRO-seq, 3 replicates each, and we did a 6-day time point for good measure. So this system has been characterized pretty exhaustively over several decades. And usually the first time point that people use is 4 hours, and that's our terminal time point. And we want to see, how many waves of this transcription can we see in this relatively short time point. And just to briefly go over what ATAC-seq is. So it's noteworthy that it's a measure of chromatin structure, but it's really not a biological measure of anything, right? Like it's how accessible is DNA, and we make a lot of inferences from how accessible DNA is since the late 70s when Carl Wu was doing this in Sally Elgin's group. Like, so open DNA seems to be, is where all the fun stuff's happening, and that's what we're interested in. That's where regulatory elements tend to be. So we can use things like ATAC-seq peaks and DNA motifs to infer binding. And what I'm going to talk about a bit later is not only can we just use whether or not there's a motif and a peak, but if the peak is changing accessibility over a time course, and a motif is specifically enriched in ATAC-seq peaks that either increase or decrease accessibility, you can actually infer inducible binding or dissociation of transcription factors using that kind of information. So here's a kind of an illustration of it. Look for peaks throughout the genome. And we've already heard a little bit about 4TU-seq, but I'm going to talk about another way to look at a nascent transcription using PRO-seq that was developed by Leighton Core and Hoojun Kwok when they were in John's lab. So here's your transcriptionally engaged RNA polymerase with your RNA in green. You isolate nuclei, or you kind of puncture holes in the cells and wash out all the NTPs, and you run on these transcriptionally engaged polymerases with terminating biotinylated NTPs, which are very big and bulky, so they kind of choke up the polymerase and it stops. You shear the the nascent RNA, sequence from the 3' end, and then with strength, we sequence from both ends now, but with strand specificity, you figure out with almost single nucleotide resolution where polymerase was. And the cool thing about this is you can do 2 time points that are really close together. Say condition 1 is like 0 minutes and condition 2 is 10 minutes. You can detect genes that are activated, and you can detect genes that are repressed. You get density of polymerase in the pause region. And what other things do I want to highlight here? So, with mRNA-seq or traditional RNA-seq, to detect the gene that's activated, you have to accumulate RNAs over background levels, and that depends on how long your gene is. If you want to look at repressed genes that we heard about in the last talk, you have to think about how long it takes to decay any given RNA, and they all have different decay rates. So by using ProSeq, we can look at immediate changes in transcription. So here's the data for those of you who like to look at genome browsers. Just to confirm the data is good. I always, we think a lot about QC metrics, but sometimes just looking at the data in the browser is QC enough. So here are some ATAC-seq peaks. Here's a gene. There's about 200,000 ATAC-seq peaks, depending on where you cut thresholds, 30,000 genes. And we're only interested in those ATAC-seq peaks in those genes that change over the time course, over the 4-hour time course. So we can take all those 200,000 ATAC-seq peaks, about 30,000 are dynamic, and we can start clustering those into any arbitrary number of clusters. And here I'm showing 5 clusters. So you see each one of these red traces is an ATAC-seq peak that increases accessibility over the first 4 hours. Each one of these blue traces is an ATAC-seq peak that decreases accessibility over the 4 hours. You can see the number of traces in each cluster here. So we can take all these peaks, we can look for overrepresented motifs underlying these, and we can do de novo motif analysis, so we don't have any a priori knowledge about what sequences are in databases. So then we match these sequences to databases, and we get, well, this is great. This is a positive control. We added something that activates AP1 family of transcription factors, and we get the AP1 motif out. We added a glucocorticoid, we get a GR motif out, and we can do this exhaustively. And we're only looking for motifs that are specifically enriched in those that are increasing accessibility or decreasing accessibility relative to those that are not dynamic over the time course. So I'm not showing the ProSeq, but in addition to ProSeq measuring transcription, you can look at changes in bidirectional transcription to infer regulatory elements that way. So you get about 14 different motifs out from all these different transcription factor families. And they are indeed just motifs. They are not transcription factors themselves. So be wary when you match to databases and something says, June or something, like it could be any number of AP1 factors. So this is just to kind of show that you have 6 motifs here, but here are all the different proteins that bind to these 6 motifs, and they're organized by their evolutionary conservation of their DNA binding domains. So I'll focus on glucocorticoid receptor motif right here. The progesterone, mineralocorticoid, glucocorticoid, and androgen receptor all bind to this motif here. We know that GR is inducibly binding because we added a glucocorticoid and none of these other nuclear receptors are expressed. And we can do the same kind of logic for these other factors to infer which factors are binding and changing the chromatin, because we know which ones are expressed and we know which factors are changing their expression over the time course. We're not capturing post-translational modifications that can also affect whether or not a transcription factor is active or not. Okay, so now we have a lot of different candidate transcription factors. And we've done this many ways. This might be the most intuitive way to show it. We want to first classify these as activators or repressors, or those that increase accessibility versus decrease accessibility. So we took all the ATAC-seq peaks that decrease and increase accessibility. And we took for each like SP1 motif, we'll say, we made a genome track of all the motif instances in the genome, and they were weighted by their score, so how well they conform to the consensus SP1 site. And you can see if you have a big peak around that motif, a peak of motifs around that class of regulatory elements, putative regulatory elements, then that would it means that what's happening there is you're decreasing accessibility at SP sites and twist sites. And these other factors, AP1, GR, KLF, and CVP, are associated with increasing accessibility. KLF looks ambiguous here, but we did a lot of work to show that's actually associated with increasing accessibility. Okay, so genes also have kinetics. Here's a couple genes of transcription factors, one that's activated, one that's repressed, that falls into these kinetic classes here. So now let's build out this network that we were so interested in doing. All right, so if we have a ATAC-seq peak A here that increases accessibility and really fast kinetics, and it has this motif under it, and we know, this blue transcription factor binds this motif, we can infer that that factor was inducibly bound over that time period. If gene X is nearby putative regulatory element A, and they have and there's a contemporaneous increase in both accessibility and transcription, then we can infer a cis-regulatory interaction, really relying on that covariation and accessibility and change in nascent transcription. And we can actually even make empirical estimates about how far away transcription factors can act from our data. I don't have time to get into that, but we can. Transcription factor, or say gene X encodes transcription factor X that binds to this motif, and say transcription factor X is a repressor. We can infer the binding of that repressor if we see ataxic peaks that decrease accessibility later in the time course. You can account for time delays because things have to get transcribed and translated. And if we see a local genes that are repressed and they're proximal to these regulatory elements, we can infer a cis-regulatory interaction between that regulatory element and local genes, just by using two assays, ataxic and proseq. All right, so now we can ask, well, what transcription factors, what regulatory elements are highly interconnected in this regulatory framework. So the raw data is over here on the right, for those of you who like to look at it. The network, this kind of constrained network that kind of highlights how deep the network could possibly be over the first 4 hours is on the left. And to us, it was really exciting and surprising that you could see 3 rounds of binding of transcription factors. So this is a binding of AP1 and GR to these regulatory elements that are proximal to TWIS2, and we see activation of TWIS2. We can infer binding of TWIS2 at many places in the genome. Here are 2 places that are near SP1 and SP3, and they cause repression of SP1 and SP3. In SP1 and SP3 are activators, so the repression of them by mass action would cause dissociation of these factors from their sites in the genome, which is near another transcription factor called serum response factor. We don't yet see motifs enriched in bidirectional transcribed regions or ataxic peaks for SRF, but I imagine if we had longer time points, we would. So we see 3 waves of binding and cis-regulatory interactions over the 4 hours of induced adipogenesis. All right, so TWIS2 is right here. It's also very highly connected. This is another way of showing kind of a constrained network that we can interpret. TWIS2 binds to a lot of places in the genome. If a box has a number in it, that's how many genes that are not transcription factors that are regulated directly by TWIS2 or something downstream of TWIS2. If a box has a transcription factor or a name in it, those are transcription factors that we infer binding or dissociation of later in the time course. And I guess the message from this is there's a lot of numbers that are downstream of TWIS2, so we think it's important for adipogenesis, and it's not yet appreciated as a regulator of adipogenesis. So the next step here would be to perturb TWIS2 and observe. Fortunately, others have already made the TWIS2 mutant, Eric Olson's group at UT Southwestern. So we characterized these mice, and we found that TWIS2 heterozygous mice have a near absence of subcutaneous fat. I'm not a pathologist, but I can see that these big white circles are going away in the TWIS2 heterozygous mice versus the wild type mice. And here's a phenotype that has already been characterized by Eric Olson's group, that the TWIS2 mice have reduced lipid fat or lipid droplets in the brown fat. And we reproduced this with their mice that we got from JAX. So these big white circles here in the brown adipose tissue are going away in the TWIS2 mice. So this is organismal functional data that TWIS2 is important for the process of adipogenesis. Arun also did molecular validation, knocking down overexpressing TWIS2, looking at expression changes, cellular verification in 3q3L1 cells and white adipose tissue derived pre-adipocytes. So, but by all measures, TWIS2 is important for adipogenesis. Okay, so now we have, let's see, how am I doing on time here? All right, so we have all these key transcription factors. Can we figure out what they do? They regulate transcription, but do they regulate pause release, initiation, something else? Here's another way to kind of, we have these networks are so vast, we can organize them in various ways. Here's a way that we can identify genes that are, or visualize genes that are exclusively regulated or predominantly regulated, because these are all inferences, by a single transcription factor. So let me walk you through it. So these are 4 activating transcription factors in our network. All these regulatory elements here are bound by GR. These regulatory elements are exclusively bound by GR. These are also exclusively bound by GR. These here, this 2,800 and these 1,400 are bound by GR in some combination of KLF, CVP, and AP1. These are terminal regulatory elements, no evidence that there's changing transcription nearby at a gene. And these are linked to downstream genes. So we have 1,100 genes that are probably regulated by GR in some combination of KLF, CVP, and AP1, but we have 174 genes that are regulated predominantly by GR. The numbers are probably not going to be exact, but like predominantly regulated by a certain transcription factor. And people have actually been coupling single treatments to nascent RNA profiling for over a decade to figure out what transcription factors do. And the original work was done by Nass and Ha when she was in Lee Krause's group, treated cells with estradiol, looked at estrogen responsive genes, and looked at the change in polymerase density in the pause region, the gene body, and came to the conclusion that ER acts at initiation or prior to initiation. Charles Danko, when he was in Adam Seipel and Lee's group, Lee Krause's group, treated cells with TNF alpha for very acute time points to activate NF kappa B, and came to the conclusion that NF kappa B acts at elongation. Fabiana Duarte, when she was in John Liss's group getting her PhD, showed that HSF acts at this pause release state. Stuff that we've done where we've tagged our favorite transcription factors with degrons and just depleted one transcription factor at a time, we showed that ZNF143 acts at initiation. So these are all one-offs, right? So we know a lot about few transcription factors, and we know a lot about the few that are rapidly inducible. But now we have a kind of a regulatory cascade where we can have a bunch of different genes that are predominantly regulated, or we can conclude that they're predominantly regulated by a single factor. So we can do these same kind of experiments and open the floodgates for the number of factors we can study. But first, I'm in a systems biology center, so I want to show kind of a model of maybe getting more quantitative view of whether or not a factor is regulating something like initiation, pause, release, or elongation. So we have empirical measurements from ProSeq of densities in the pause region and the gene body region. And we know the different steps that have to take place: initiation, premature termination, pause, release, and elongation. And we can look at the changes in these kind of composite profiles, either for all those 174 GR responsive genes, or we can do this on an individual gene basis as well, and ask how is the densities changing in the pause regions versus the gene body between time points. We can formulate this into ordinary differential equation models where we have empirical measurements of pause density, gene body density. We can model them at steady state and ask what rate constants for initiation, premature pause release, and pause release could account for the densities that we see in the gene body and the pause region. Calculate it at steady state and ask how would you change those numbers, those rate constants, to account for the differences that we see in gene body and pause densities. So here the 170, this is the composite profile for the 174 GR responsive genes. And even though a lot of rates for initiation and elongation and premature pause release can account for the initial steady state, only by changing the pause release rate by a factor of, say, like 1.5, can you account for the change in profile that we're seeing, and really modest changes in initiation. We're holding elongation constant and premature pause release constant for reasons that I'm not going to get into. So we can do it for the average. We can get if say we assume a rate of elongation of 2.5 kb per minute, we can actually get estimations of residency time in the pause region and how that changes between 0 and 20 minutes of induced adipogenesis. The polymerase being there for 30 seconds and going down to 20 seconds when you increase pause release rate by 1.5. So this was, these are all inferred GR binding sites and inferred regulatory events that are predominantly regulated by GR. So what we did is we took some acute lymphocytic leukemia cells that were isolated many, many years ago. We treated them with dexamethasone for an hour, and we looked at the change in composite profile for those responsive genes. And we see a very similar thing, that GR tends to be regulating pause release. We can only account for the differences in gene body and pause densities by changing the rate of pause release, and rates of initiation won't really affect. We can't account for our empirically observed densities. A little bit different residency times that we calculate if we assume an elongation rate of 2.5 kb per minute. We do the same thing for other factors like SP1. We have a little bit more genes here. SP1, it's one of the earliest characterized kind of promoter bound sequence-specific transcription factors. Although it wasn't really characterized at the time, they didn't know SP1 was binding there, but we show, and others have shown this as well, that it predominantly affects initiation. So we can only account for the changes that SP1 targets by initiation. And we recall SP1 is dissociating, so it's reducing initiation rate. And we can even get an estimation of the number of molecules per minute that are changing between these two time points. And SP1 is activated later in the time course. Okay, with that, I would really just like to thank my whole group, which is a great bunch of scientists, great bunch of people interested in transcription, and predominantly Arun, who's been working on this project for the past 4 years. Be happy to take any questions.
Awesome. Thank you, Mike. That was awesome. A reminder, you can either type your questions in the Q&A or raise your hand and ask them yourself. I have several, so maybe I'll just get started. I was wondering for what you're seeing with kind of the interesting regulatory elements. Most of what you talk about were things where they're changing and exhibiting changes in chromatin accessibility. Do you think that's largely what's happening in the system, or do you also see some where the open chromatin is preformed and primed for transcription factor binding, and then you'd see stable accessibility within a change in nascent transcription?
So I think most things, if say your predominant role as a factor is to recruit general transcription factor machinery or polymerase, so you set up these bidirectional promoters. I think that ultimately results in higher accessibility of the region. It's not maximally accessible, and so there's always a little bit of dynamics, so it can increase accessibility a little bit for things like factors that are involved in nucleosome remodeling. I imagine that there'd be bigger changes in accessibility, but even things that are involved in pause release, right? We see GR, we see GR motifs specifically enriched in those that increase accessibility. And I think that's just because more things have to get in there. You're recruiting more things, it's more accessible. I don't know if that fully answers your question, but ATAC-seq seems to capture almost everything that so all the regulatory elements that we define by bidirectional transcription, not all of them, but let's say the motifs, we also find those by ATAC-seq. But there's technical reasons for this, like most of the pro-seq signal. So if you sequence ATAC and pro to the same depth, right? Most of your pro signals are going to be within gene bodies. You're not going to have the effective read depth that regulatory elements that you would have for ATAC-seq, where 60% go to peaks. So I don't know if that fully answers your question, but I think for defining regulatory elements or putative regulatory elements, I think ATAC-seq works pretty well, even for things that recruit initiation factor machinery or things downstream of that.
So you're largely seeing, even if it's the accessibility is there already, you're largely seeing a change that you can measure.
Oh yeah, that's a good point. I mean, that goes back to the whole pioneer factor thing. It never goes from inaccessible to accessible. We're always basal level of accessibility, and there's a concomitant increase. I mean, I'm sure there's several, many places in the genome maybe they're at maximal accessibility and we're not seeing the change. But in genomics, you're looking at the whole class of things that change. So maybe I can't really put an estimate on it, but we're saying GR is binding to a bunch of different sites. There might be sites that are really already very, very, very accessible. GR is binding, but there's no concomitant increase in accessibility, and we're not observing that. So we are limited. I think we're limited for any individual. Our way of inferring these networks is pretty specific, but it's not necessarily sensitive.
Yeah. And then one other question is, on the GR with it regulating pause release, can you speculate as to what are you thinking about for the mechanism of that?
I can speculate that it recruits PTFPs directly or indirectly. I don't know exactly what's going on.
Awesome. Okay, so Joseph has his hand raised, so you should be able to unmute yourself and ask a question.
Okay, can you hear me now?
Yes.
Yes.
Okay, great. No, thanks. Yeah, that was very, very interesting. So I'm curious about whether you're thinking about or how you would like functionally validate some of these this like graph, this bipartite graph, like the relationship between which TFs are binding to the regulatory elements and which of those regulatory elements are regulating which genes. Maybe it's like imagine that you mentioned this covariation type method for predicting these things, and yeah, I guess it's just like not clear like how well that's working and how you would think about validating
So
it if you
I
want
would
to.
definitely hang my hat on some of the conclusions that these certain factors that are showing up again and again are specific, but any specific interaction, right? Like I don't know how to, if you have a better way of figuring out whether or not a regulatory element is actually affecting transcription of a local gene besides doing the editing there,
Yeah.
and doing the induced adipogenesis, which won't happen anyways because you do the edit at that site and these things can't differentiate anymore. Like I don't know. We can't do that at each one of these individual things that but I would still argue that this is the best way to do it. We have time points on the order of 20 minutes, right? Like if you have a regulatory element increasing accessibility and a gene increasing transcription, they're proximal to one another in this short window. I don't know how many other conclusions you can make except that one's probably regulating the other. But yeah, we can't validate each one of those. And I'd say the chip so obviously one could do chip to validate the binding and we can look at these things. But I would actually argue, chip is not it measures transcription factor binding. It's not sensitive. It's not specific. It's only specific as your antibody. People don't do all the proper controls where they, knock down their factor and also do the chip. So, there's all reports of tethering all over the place, which is, not real. So I would argue that in terms of specificity, if we're inferring binding because we have a motif that's specifically enriched in one class of regulatory elements that's changing direction, and we have an accessibility peak that's changing in that direction and the motif is there, I would say like I'd be more willing to hang my hat on the transcription factor binding there than I would any random chip peak that is defined by some arbitrary threshold. So we can use, so the answer is we can use chip to validate it, but I don't know if it's a better orthogonal or worse way to infer binding.
Yeah. I mean, yeah, thanks. I don't have a better way to validate these connections except for doing like the the CRISPR experiment or something like that, and that's tedious. You can't do it for all of them. I guess
it doesn't matter really sometimes.
Yeah, I mean, I guess it's almost like what you're actually going after. And I guess I would say like, this sort of covariation analysis, like, if there's like one peak and there's like one gene and that peak is like a KB from the gene, it's like, it's in the gene desert. Okay, fine. But like, a lot of times in the genome, there's like very, gene dense regions where there's a lot of these ataxic peaks and a lot of these genes like, all within like, tens of KBs of each other. And that's, I'm not sure if that's what you're observing, but like.
Well, I'd like to interject there. Yes, there are hundreds of thousands of ataxic peaks. We are relying on a change in accessibility, right? Like in a short time period. So that is what's increasing our specificity, right? Like we're not just saying ataxic peaks there regulating that gene, right? Like there's a change in accessibility. So it's either coincidental that like you have a change in accessibility, a change in transcription at the same time point, in the correct direction, increase accessibility, increase transcription. Like that's either a coincidence or there's probably some regulatory potential. I'm sure like we're not, I don't know how sensitive or specific this is. It's really hard to determine that, right? Because we don't have en masse ways to determine, whether or not a regulatory element's important. Sometimes they're redundant. They could be redundant, right? Like, so I don't know. So yeah, it's not just having a taxi peak. There's a change in accessibility. They're nearby, and that's what's kind of driving these cis-regulatory inferences.
Yeah, gotcha. Yeah,
thanks.
Maybe one other question. I was like, so you see reports about this sometimes about how like certain transcription factors, they'll have multiple motifs within a region, and that that can be important for their function. Have you looked at that? Like, are some of these kind of key cis-regulatory elements that you're finding, do they have a single motif, or do they have multiple motifs? And does that tell you anything?
So many of them have multiple motifs. I can maybe toggle back, this regulatory element here will have a GR motif in one combination of these other factors. So it could maybe the 350 of them have KLF and all these numbers are associated with it. So those are the motifs that are changing accessibility, those are the motifs that are present that are driven by factors, the factors that recognize those motifs are, increasing accessibility over the time course. But that's not to say that there's not another motif there that's like already, predetermining whether or not a factor can bind. It could be maybe like FOXA1, GATA3 or something that's already there, but but a GATA3 FOXA1 isn't activated or increasing or decreasing expression, so it's just maybe kind of constantly there. So you see that motif kind of at the same levels in those that are non-dynamic versus increasing or decreasing. So yeah, we see multiple motifs there. We've looked at kind of the cis regulatory like logic. Do factors, just having two motifs of different factors, do they synergize, or are they kind of completely independent in terms of whether or not they're going to regulate a proximal gene? So we have these analyses. I think like GR and AP1 tend to synergize, but like in this network here, twist1 and SP, they seem to be kind of operating completely independently. Like if you have a twist motif, an SP motif, like if you have both motifs there, you're no more likely to regulate a nearby gene than if you just like randomly have a twist motif and SP motif being present. Yeah, so we looked at that a bit, but it's not really clear.
And like for like say like just the SP motif, for instance, do you see multiple SP motifs within a regulatory element, or just the one? So I know some of your motifs are like double peaks, and those could be because they're in different places and different elements, or it could be that some elements actually have multiple.
Yes, so we do see several peaks, right? Because the taxi peak, I think we define the window as 200. So it might have two GR or two SP motifs, and I don't think we discriminate and say that like GR is bound to this one but not this one. So we've done all those kinds of analyses where you say like having multiple motifs for several transcription factors. Just having more motifs influence whether or not you're going to have a cis edge drawn, and it's actually not the motifs that are driving it. The number of motifs. It's actually what's driving that is the change in accessibility. So if you see a big change in accessibility with like one motif, it's more likely to be an edge. Like, so you're more likely to have a gene nearby that's increasing transcription than is if you have like, 14 motifs and a very modest change in accessibility. There's less likely to be a change, because I think that the accessibility is more the functional consequence of things binding. So like in all these things, right? Like, taxi peak, they span orders of magnitude. Like, a really small ataxia peak is probably more like a non-ataxia peak than a big one. So likewise, a very small change in accessibility is probably more like non-dynamic versus a really big change.
Yeah, that makes sense. Okay, great. Well, we're 5 minutes after, so I think we'll close it up there. Thank you again for speaking. That was fantastic. Thank you everyone for coming, and reminder, we'll be back January 18th with talks from Olitz and Jacques Cote.